Hello, everyone. Thank you for joining us tonight. We'd like to welcome you to the United States Geological Survey Public Lecture Series. My name is Christy Ryan, and I will be your host and moderator today. Before I introduce our speaker, I have a few announcements to make and allow folks out there a little bit of extra time to get logged in. First, I'd like to give you a heads up about our lecture for next month. On March 30th, we will have the pleasure of welcoming Ilse Kuffner with our USGS St. Petersburg Coastal and Marine Science Center. The title of her talk will be Coral Reefs in Crisis, Science to Guide Reef Restoration for Ecosystem Recovery. So please be sure to save the date and join us next month as well. At the end of the lecture tonight, I will provide information on how to join our mailing list, so you'll always be in the know on upcoming lectures. Next, we want to just take a second to give you some tips on a few features of this Teams platform. One feature we want to direct your attention to is the question and answer panel, which can be opened by clicking the question mark icon in the upper right-hand corner of your screen. At the end of the lecture, we will have a Q&A session, and this is where you can submit your questions for our speaker. Just a heads up, we'll do our best to get to all the questions. We hope you can understand if we can't get to everything. Another feature we want to give you a heads up on is the closed captioning. If you're watching on a desktop computer and need the closed captioning, just look in the bottom right-hand corner for the closed caption icon, or you can use the stream text link that we provided in the Q&A panel. Okay, and now, I think that's enough of all that business. I want to introduce our speaker for this month. Dr. Kimberli Miller has been a wildlife disease specialist at the USGS National Wildlife Health Center since 1992, working on wildlife disease issues and problems across the country. This involved providing technical support to state and federal natural resource management agencies, conducting field investigation into Newcastle disease and white nose syndrome, and participating in a long-term whooping crane reintroduction project. Currently, as a member of the center's informatics team, Dr. Miller is actively involved in data management and designing the center's data systems for ease of use and better data availability. She earned a Doctor of Veterinary Medicine degree and a BS in Animal Science from the University of Missouri-Columbia, and is thrilled to have found a way to combine non-traditional veterinary medicine with large-scale wildlife conservation. I also want to highlight that Dr. Miller was featured by the Department of Interior's Career Group for her work as a veterinary medical officer and wildlife disease specialist in honor of Black History Month. So without any further ado, let's all give a warm virtual welcome to Dr. Miller. Kim, the floor is all yours.
Great, thank you very much. I'm so thrilled to have this opportunity to spend a little time talking to you about the National Wildlife Health Center and what we do there. And I'll talk a little bit about disease control and whether there's things we can do about wildlife diseases. And I'll also talk about why it's important that we care about wildlife health. First, though, I wanted to give you a little bit more background on me. I love to travel, and my family and I like to get out and explore, and just especially getting out into nature and seeing new areas. And I've always loved science, and I've always loved animals, particularly wildlife. And I think in part that could have been from watching Wild Kingdom as a little kid on Sunday nights. So I think something in watching them out working with wildlife sort of triggered that interest in me. So when I was in vet school, I had the opportunity to spend 6 weeks as an extern at the Lincoln Park Zoo in Chicago. And that honestly was the best 6 weeks of vet school. I got to see all kinds of interesting things and do all kinds of interesting things. And it really helped me to understand that I wanted to work with wildlife, but not so much on the clinical medicine side. I wanted to work more on the wildlife conservation side. So after I graduated, about a year after I graduated, I landed my dream job at the USGS National Wildlife Health Center in Madison, Wisconsin. And so for the first 10 years, I was a wildlife disease specialist there, one of the epidemiologists who had a section of the country that I was responsible for, helping the people resolve their wildlife health issues, questions, problems. And then for the past 20 years, I've worked on a variety of different projects. So for the next 45 minutes, I'm going to talk to you about the center and what we do and take you behind the scenes of a wildlife disease investigation. So for disease to occur, you need three things. You need a susceptible host, you need a disease-causing agent or a pathogen like a bacteria or virus or parasite, and you need an environment that brings both of those things together, that both those things can live in. And so that's what this little diagram is on the screen by the turtle. It's that intersection of host, disease agent, and environment that creates the conditions for a disease to occur. So wildlife disease investigation is a very collaborative process. And so although I'm the person here talking about the center tonight, I'm part of a much bigger team of people that are working together to sort of figure out things about wildlife diseases. Because there's so many different people involved and different agencies involved, then communication is really key. It's very important that we can share information back and forth. When we are coming up with answers and figuring things out, it often leads to more questions. And lastly, ecosystem health, animal health, and human health are all interconnected. So those are some of the themes that you'll hear me talk about over and over this evening. So when I take you behind the headlines, we're going to walk through the systematic process that we use to determine the cause of death and figure out what's going on and whether there's something we can do about it. We'll explore the importance of healthy wildlife, and at the end, I'll share some resources in case you want more information. So when wildlife make the news, particularly when they're sick and dying, it's because they are dying in alarming numbers. For example, this headline from the San Francisco Chronicle in 2020 was about an avian botulism die-off that killed 40,000 birds and was threatening millions more at Klamath National Wildlife Refuge. Sometimes they make the news because it may be a new disease, an emerging disease that's having significant population declines. For example, frogs in Minnesota. This article from the Duluth News Tribune is about frogs in Minnesota and elsewhere that were dying of a new disease, and it was really wreaking havoc on populations. And more current is this headline about avian influenza that is being detected now in minks and sea lions and other mammals, and trying to figure out what the significance is of that. So for the next few minutes here, I'm gonna talk about a disease that first made headlines in 2008. So this is an article from the Daily Gazette out of Schenectady, New York. And it's about a mysterious affliction ravaging bat populations that was first detected in New York. And this headline is about how it's now been found in Vermont and Massachusetts. So the one thing that all of these headlines have in common is that before there can be a headline, someone has to notice that there's a problem. And they have to communicate it to someone that can do something about it. So that's really the first step that gets the ball rolling. And that's what happened in the winter of 2007 and 2008. People in New York started reporting seeing bats outside in the winter. They were being found in people's backyards. They were clinging to the sides of their house. And it wasn't just one person reporting it. It was lots of people. And they weren't just talking about one bat, they were talking about a lot of bats. And so this picture, this in the center of the screen, it looks like it could be a leaf or a piece of bark, but it's actually a bat that's out crawling on the snow. And that is highly, highly unusual, because once bats settle in for the winter in their hibernaculum, they don't leave. They don't leave until spring. So to see bats out in the snow that's unheard of. But that's what people were reporting. And so those reports then often are going to the state natural resource management agencies. And I wanted to talk a little bit about sort of the state and federal agencies. So states have the legislative authority to manage public lands and wildlife within the state. And so they all have slightly different names. In Wisconsin, it's the Department of Natural Resources. In New York State, it's Department of Environmental Conservation. Vermont is Vermont Fish and Wildlife Department. So they all have slightly different names, but they kind of do similar things. They are responsible for hunting, fishing, other recreational activities. They manage public lands, so like state parks and county parks. They have all kinds of educational programs, and there's just a whole wide range of things that they do. And they work a lot with animals that kind of stay in, they don't really migrate, they stay within the general area. So deer, raccoons, squirrels, those kind of animals, those come under state stewardship. And that's as compared to federal agencies like National Park Service or Fish and Wildlife Service or the other agencies under Department of Interior. So at the federal level, we work with migratory birds, endangered species, and animals on federal lands. Those all come under federal stewardship. Now, there's a lot of overlap there because certainly the states do a lot of work with migratory birds. And so there's a lot of collaboration that happens between the state and the federal. But for those initial reports, when people are seeing a problem, then those get reported to the state game agencies. And that happens quite frequently, where it's the public that first notices something and is reporting it. We've seen that time and time again with different die-offs and different diseases. So that's what happened here. The public started seeing birds flying in the snow, and they reported it to the New York biologists there. They also reported it. They started getting reports in Vermont and in Massachusetts. And so all of those biologists were getting reports from people saying, hey, we're seeing bats outside. What's going on? So those folks, now the person here is the biologist in Vermont, and he's at one of the sites where bats were affected. And so at the state level, then they're communicating with the public. They're also communicating it back and forth with other biologists. So checking, with biologists in other parts of the state, in neighboring states. Are you seeing a problem? What are you seeing? This is what I'm hearing and what's being reported to me. But they're also then communicating at the federal level as well. And so they're getting in touch with the center. And here's the current epidemiology team at the center. And so they're sharing information back and forth. And from the epidemiologist side, we're asking the same sort of questions that you would be asked if you took your pet to the vet. So things like, when did you first notice there was a problem? What are the sick animals doing? Are there animals there that are in the same area that aren't sick? How are animals using this site? We're really trying to get a good history, a really good understanding of what is happening and what's going on, because all of those things help us get a better picture of what it may be. So the public was talking with the state folks were talking among themselves and also talking with the federal folks. And we were in contact with other state people checking around, like, are you seeing, what are you hearing about? Because we're getting these reports. And so that's one of the benefits of our lab being at a national level and working all over the country. Okay, so step 2 in this process is samples need to get to a laboratory for testing. Now, this is not a bat, it's a bird in that upper picture, but the process is the same. We need fresh, dead, chilled carcasses to work with. We can't really do much with something that's rotten, so we need it fresh, chilled. And in the winter, that's a lot easier because it's cold. In the summertime, it's more challenging because things decompose really fast on a hot summer day, especially something small like a bat. So what the person in the picture is doing is they've put a tag, an identification tag on that bird, and they've triple bagged it. They've put it in a cooler. They've packed it with ice packs, like the blue ice packs you get from the grocery store, to keep it chilled. And then they shipped it overnight delivery to the center. And so that's coming into here. This upper building is the main building of the main diagnostic lab for the USGS National Wildlife Health Center. This lower picture is our research facility there on the same campus. We're based in Madison, Wisconsin, but we really work all over the country and internationally as well. So we are the nation's only biosafety level 3 diagnostic and research laboratory in the country. And so we're really rather unique, and we're really rather unique in the world, actually. And so our whole focus is on disease prevention, detection, and control, and examining wildlife health, looking at trends. We have a rapid response team if we need to go out to a site and do a field investigation. We do research. We do a whole wide range of things in support of wildlife health. So as I said, we're based in Madison, Wisconsin, but we also have a field station in Honolulu, Hawaii. Our mission is advancing wildlife health and science for the benefit of animals, humans, and the environment, and our vision is providing new solutions to promote wildlife health and ecosystem resilience. And really, the center started in the 70s, actually, when there was a really large die-off of over 40,000 ducks that died at Lake Andes National Wildlife Refuge in South Dakota. And after that die-off, the U.S. Fish and Wildlife Service asked Congress for funding to start a national lab because they needed a place that could do the diagnostic work, research work, and provide disease control assistance and management information when there was a die-off, a wildlife die-off. And so that was really kind of how the center started. And so you can think of us as your federal tax dollars at work. And it takes a large number of people. There are about 100 people altogether that work at the center, and it's a wide range of specialties. So we have folks that, we have scientists that study bacteria and parasites and viruses. We do some toxin work at the center. We do disease discovery. We have folks that work with data a lot and look at disease trends and data science. We have veterinary pathologists on staff who not only do they have a degree in veterinary medicine, they've gone on for additional training and are board certified in pathology. We have animal care techs, and we have lab technicians. There's a whole suite of folks. So it really does take a village to try and understand these diseases, and that's really one of the best things about working at the center, and in fact, in working in wildlife in general, because in general, people that work in wildlife are very easygoing. They have a great sense of humor. They're smart, they're dedicated, they're just a lot of fun to work with. And if you have to work on something challenging, you want to do it with people that are enthusiastic and fun. So my colleagues at the center and our partners in the field are really one of the best parts of this job. All right, so I explained how the biologists in the field are out, they collect the carcasses, they've talked to the epidemiologists at the center, and we've gone over like what kind of carcasses we want, how many, how to package it. We talked to them about wearing personal protective equipment when they're handling sick and dead things, because safety first is really uppermost in all of the things that we're doing. So that cooler of animals came into us overnight mail and get delivered to our dissection suite. So this upper picture is showing a number of coolers that have come in from all over the country. That's the shipment that came in that morning. And they are then, there's carcasses in there that will be examined. This is our dissection suite, and it's because we work with a number of different diseases, which I'll talk about more in a little bit. We have special air handling systems in that facility. There's special water treatment systems. There's an incinerator right there. We really take safety seriously. So there are a number of safety and biosecurity features and processes that are built into what we do, both literally built into the building and then in our processes. And that's why, I mentioned that we are a biosafety level 3 lab. And what that means is that we can work with some very serious diseases that could be potentially deadly for people. And we don't want that to happen. So we have all of these processes in place to protect ourselves from that. And also, we don't want to transmit or take diseases outside of this space. And so that's another reason why we have all these layers of protection. Now, you notice in this picture here, the people in the picture are wearing all kinds of garb. They have white Tyvek disposable suits. They have rubber gloves. They have plastic head covering. They're wearing masks. And that, again, is all part of the personal protective equipment and safety, biosafety aspects of what we're doing. In fact, to even come out onto this floor, there's a whole process. First of all, there's a training that you have to do, and you have to pass the quiz with that. And then to go from the locker room out to this floor, you have to completely get out of what you're wearing, put on a robe, go down to a different locker room, change into scrubs, rubber boots, and get all garbed up with the gloves and the disposable coveralls and all of that before you can come out into the space. And depending on the disease that we're working with, if it's something that has a high human health risk or has a domestic animal, a high domestic animal risk, then they have to shower out. So after they leave the floor and finish their work, they go directly into the locker room and they have to shower and wash their hair before they can proceed any further. Again, all the steps that we're taking because we do work with some serious diseases. So step 3 is a diagnostic evaluation. And so a necropsy is like a human autopsy, and it's examining the veterinary pathologist. They're examining these animals inside and out, from head to toe. And so they're starting with an external exam. You look at the nose, the eyes, the ears, you're looking at the skin, you're examining outside, are there parasites on the skin or on the feathers? You're examining all of the things from head to toe. And then you do the same thing, they do the same thing internally. They're looking at the mouth, they're looking at the lungs, the heart, the liver, the intestines, is there food in the stomach? There's all of these things that they're looking at. And it's a very systematic process that they're going through. And based on what they're saying, when they do their external and internal exams and the history that we've gathered, all of those things are helping them formulate an idea of what might be happening, what might be causing these animals to be sick and dead. And so they're kind of generating a list of things that they're trying to rule out or rule in. And one of the challenges that comes along with doing diagnostic work on wildlife is that there are so many species. In 2022, the center received 158 different species of animals. And so for the pathologists to understand if something's abnormal, they have to know what normal looks like. And so that's a lot of different variety that they have to be familiar with and know so that they can understand what is looking normal and what's not. So based on all of those things, the history, what they're saying, they're deciding at the table what samples they need to collect and what tests they need to run in order to get a better understanding of what's going on. So they may be taking tissue samples and sending them up to our labs for folks to check for bacteria, fungi, viruses, parasites, toxins. The histopathology means that they're taking samples of tissues and putting special stains on them so that then the pathologist can look at them under the microscope and look at the cells and see what's happening at that level. So with this bat disease, one of the things that was first very apparent is this white stuff around their nose, and that's how it got its name, white nose syndrome. Well, it turns out that white stuff is a fungus. And so that was another unusual thing to find. And that it highlights one of the challenges that we see in trying to do diagnostic work, because it's hard to grow things in a lab sometimes. Even things that are incredibly deadly for wildlife can be a real challenge and difficult to grow in the lab. And so for this disease, this fungus, at first when the lab folks tried to grow this fungus and incubate it, they found that they had to incubate it at a lower temperature, at a colder temperature than they typically would have to with other fungi. And so it's kind of learning, it's trying things, learning things, applying what you know from one area to another area. But it takes time. And I think that's something I know is challenging. I know in the movies, it seems like it happens really fast. Like there's a die-off and then boom, 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 there's some tests and voila, they have the answer. But in real life, it's a much longer process because there are these challenges that we're having to overcome and figure out. And so it's really, all of these pieces of information are pieces of a puzzle, except when you're dealing with a disease that's new, that's like putting together the puzzle without having the picture on the box. So another step in the process is a field investigation. Now, we don't do field investigations for every die-off that happens, but we definitely do if it's a new disease or something that's highly unusual. And so bats flying outside in the winter was definitely unusual. And so the center sent me out on a field investigation. I went to visit the sites in New York, Vermont, and Massachusetts. And so I have a few slides, I'll talk about that. The reason that we do field investigations is we want to know more about this disease, and we want to learn from the people right there on site, because they know more than anyone about all the intricacies of that area and the species in that that are involved. And so my first stop was into Vermont, and as I mentioned, safety first. Whenever we go out in the field, we pack along supplies. So that's what the cooler in the upper picture is. I took my boots and gloves and disposable coveralls and all the equipment that I would need to collect samples and disinfectant, so I could disinfect things between visits. And we went out, this was late March, we went out to Aeolus Cave in Vermont. So approaching this, in these crevices of the rocks, there were bats tucked up in there, and they should not be there. They should be deep in that cave. There were also dead bats on the ground there. So in this picture on the left, I'm in the cave now, and I'm looking back up toward the entrance. There's a biologist that's coming down as well. And to my back are these metal grates in these two pictures on the right. And because this cave, well, prior to this die-off and event, this was a hugely significant cave for bats in the Northeast. So this was the largest bat hibernaculum in the Northeast. There should be thousands of bats in the winter in that cave. And so because it's such an important hibernation site, they had blocked it off so that people couldn't get in there and harass the bats. So to get in, the biologist had to remove one of the slats, and then we had to crawl through to get in. And near the gate, there were these ice mounds, and there were bats clinging to the ice. Again, they should not be doing that. And it's hard to see, but over here next to the ice, there are dead bats. And going deeper then into the cave, there were thousands of dead bats. I mean, it was heartbreaking because there are just so many. And there were bats clinging to ice. And there were still some live bats in that cave. You can see in this picture where there's bats overhead. With a disease that's easily spread from bat to bat, you can see how easy it would be to spread quickly through this population because they're all packed in together. But when we looked at some of the bats, you can, in this wing, there's a dark spot in the wing and it's very crinkled and dry. It should not look like that at all. And then there's a very light area around it. And so the wings should not be all dried and wrinkled like that. They look very dehydrated. And that's a big problem for bats because their wings are hugely important, not only for flying, but they also are important for managing their blood flow and their blood pressure. So it helps to, the blood vessels in their wings help with managing blood flow to the heart when they're hanging upside down. There's also, it's very easy for them to get dehydrated. They can lose a lot of fluid across their wings. And so damage to the bat's wings means it's really interfering with their normal physiology. So one thing that you'll hear in science is the importance of publishing. And the reason for that is because science is cumulative. Science builds upon each other. And so the knowledge that is gained today can be used by someone tomorrow. And that's what I wanted to highlight in this slide. So this is the outside of that cave, of the Aeolus Cave in Vermont. And this dot in the map is Aeolus Cave. In the 1950s and 60s, there were some biologists that banded bats. And so just like with birds, they put little bands on them. And then the people, when they found those bats with the bands, they would report where they were being found. And so we knew from work that had been done in the 50s and 60s that come spring, the bats in this cave would go to multiple states. They would disperse there in the Northeast. And so if this was a disease that surviving bats could carry with them, then the probability was high that this disease was going to spread. And that's what happened. So from Vermont, I disinfected all my stuff. I went on to New York. New York, we were going into a mine. It was a little bit warmer that day that we went in there, although this patch out here of snow and ice was rather slick. You can see that we're going in between this sort of cracks in the rocks, and that people are wearing disposable coveralls, boots, hard hats. There's 2 headlamps for everyone, and they all have fresh batteries, which will become important in a minute. And so in working our way then through those crevices, we came upon this hole in the rocks. And so this is my right knee. This is my boot over here. This is my left foot. And this is a person's leg that's already in the mine. And so they said, okay, Kim, just drop down through that hole. It's like, okay. All right. And that's what I did. And I'll have to say, this was one of the most interesting and surreal experiences I think I've ever had. Because when I dropped down through that hole, this is what I saw. It was very dark in there, very dark. And that's why you wanted to have fresh batteries and 2 headlamps. So in trying to get oriented, I discovered a couple of things. One, the headlamps shine, they're up on top of your hat, they're shining straight ahead. So you can't just glance down with your eye, you have to move your whole head down if you want to see your feet. So I was, trying to get oriented and figure out how I'm going to navigate down. The feeble flash from my camera couldn't do it justice, but you can kind of get a sense how steep this was. Here's a person up here, and here's some other folks that had traveled into the cave with us down here. And this was a lot of loose gravel and some large rocks. And you can see from the pants of this person that sometimes it was easier to just sit and scoot down. The one thing I could notice was that down at the bottom, there were some very large rocks. This is a full-size backpack, just for comparison. And so I was being particularly careful that I did not want to slide down that hill and end up in the rocks, because I did not want to see a headline, "Klutzy federal scientist falls down in mine." That's a headline I don't want to see. So anyway, we all got down safely. And we were navigating around in this space. And I just wanted to include this other photo. Here's 2 pictures of 2 of the people that came in that day. They were other biologists there from the state. And just to get a sense of how big this space was, how big this mine was. And actually, one of the things that they were doing was they had taken, they were going to be taking photographs of the walls, and because they wanted to, it was how they found the best way to try and count the bats in that cave, was to take photographs of the bat colonies and then take it back to the lab and count the bats in the photographs. So after climbing through the bars in Vermont and dropping down through the hole in New York, I wasn't sure what to expect in Massachusetts, but we were also going into a mine there, but we just walked right in. And there was water in the mine, but it was not over my boots, so that was fine. We were able to just get in and get the samples that we needed. And I just wanted to highlight that there are a lot of challenges sometimes with doing field investigation work, that, there's a lot of benefits to it because we get to see things firsthand for ourselves and collect samples. We get to meet with all the different folks. It's a good opportunity to get all the people and agencies involved together in one spot and sort of talk and plan through what needs to happen next, and help them with disease control and disease management, talk about personal protective equipment. Sometimes we end up doing interviews with the media to help that way. But there are a lot of logistical challenges sometimes with doing field work. It takes us into sometimes very wild areas, sometimes very remote areas, because that's where the wildlife are. And so this larger picture here is our colleague, Dr. Terry Work, who heads up the Honolulu Field Station, and he is out doing work at Palmyra Atoll in the Pacific. It's roughly in the middle of the Pacific Ocean. And this site where he is, it's not inhabited year-round. So the logistics of trying to get people and supplies and all of the things that you need to the middle of the Pacific Ocean is quite a challenge. But we have to be creative problem solvers and adaptable. So for example, my colleague here is using the back of a pickup truck as a workbench, and there's a tarp overhead to keep the rain off. So again, we have to adapt and keep rolling with it. So really the scientific method, you're making observations, you're coming up formulating questions, you're figuring out, how to test those things. What samples do we want to collect? What do we need to do? We're running the tests and samples or experiment. We're coming up with the conclusions. We're generating reports. We're getting the information back out to the field and trying to determine, what we can do. Is there something we can do about this? Now, as I showed you this slide at the beginning to talk about for disease to occur, you need a host, an agent, and an environment that they both can live in. But I did want to point out that there's a difference between disease diagnosis and pathogen detection. And that's the case with the bats. So this white fungus around the nose, it's very apparent, right? And it would be easy to jump to the conclusion that, okay, that's the thing that's causing it. But you can't do that. You got to dig for the facts. You need to understand if that is actually the primary cause, the thing that is causing those animals to be sick and dead, or was it just an opportunistic infection? Like if there's something else causing them to be sick and now they're really run down and susceptible, and then this other thing came in. So doing more work, looking at the bats, this picture down here, there's all these little faint spots in the wings, found that the fungus will fluoresce under black light. So that's what you can see here, all of this kind of green glowing. And here's a close-up of that. And what the pathologist found in all of the different testing and all of the different lab results that were coming through is that this fungus was the thing that was causing illness and death in these bats. The fungus invades through the tissue of the wings, and those bat wings are amazing. They're very thin. There's like a layer of skin cells, a layer with blood vessels and nerves, and another layer of skin cells. So they're very thin. And the fungus was just invading into those and causing disruption. It was interfering and interrupting normal body function. So through all of the work, then the conclusion was, yes, this fungus is what is causing illness and death in these bats. And it's a new thing. We had not seen it occurring in this country. And from doing additional work, then people did find this fungus in Europe and Asia, but it does not cause disease really in bats there like it does here. The other thing that they found is that although we first started seeing widespread problems in 2007 and 2008, there was a picture, retrospectively looking back, there was a photograph in a cave of some bats, and there was white around the nose of some of those bats, and that was in 2006 in New York. So this was a very interesting disease. Oftentimes when you have these huge outbreaks like this, you do have a lot of agencies working together. And so it will shift from kind of that initial what's going on and figuring that out into a more formal type of team, because this clearly was going to need some long-term effort to try and address some of the issues, some of the questions that came from some of the answers that we came up with. And so white nose syndrome has a white nose response team, and this is just an example of some of the agencies that are involved. Step 5 then is moving toward research, moving from the diagnostic realm and the cause of death determination to some of the longer term research questions, trying to answer, like in this picture here, this is summertime, these are bat boxes, and these boxes underneath are to collect bat droppings or guano, because they were trying to understand bats that survived, can they spread this? Do they have this fungus in their droppings, for example? This picture is collecting samples from various caves, trying to understand where is it living in the bat's environment. And this upper picture is some work looking at testing out a vaccine. Can we create an oral vaccine for these bats, taking advantage of some of their natural behaviors, because they sometimes will groom each other. And so if you can spritz them with the vaccine, then they could groom it off of each other and get the vaccine that way. So there's a number of different ways looking at trying to learn more about this disease. And with white nose syndrome, As I said, this X marks the very first spot in 2006, and then all of these colored blocks on here are where the fungus has been detected, where there have been white nose positive bats. And it's now in 38 states and 8 provinces in Canada. And there have been some additional states and provinces where the fungus has been detected, but there hasn't necessarily been bat mortality. So that all seems kind of depressing and dismal. White nose syndrome to date, millions of bats have died. It has devastated populations. Up to 90 to 100% of the bats in some areas have just been wiped out. And so then the next thing the question is there anything we can do about it? I want to talk about 3 different diseases really quick here. So avian botulism is a toxin. It's an intoxication. We see it in birds in the summertime, and it causes paralysis. So this is a bird that has avian botulism. And so because it's a toxin and it's site-specific, there's some things that we can do to try and minimize losses or prevent losses. So one thing that we'll suggest is picking up carcasses, because a carcass provides a great substrate for toxin production. So we want to get carcasses out of the environment. In some instances, if they can adjust the water levels, they can flush fresh water into an area. That can be helpful. Botulism is also a disease that birds can recover from. If they're not too intoxicated and you take them out of the environment and you give them protection from predators, food, water, shelter, they can recover. So there's a few different ways with that disease that we can respond and minimize losses. Another disease, also a bacterial disease, is salmonellosis. That is something that we commonly see in birds at bird feeders, and it's shed, it's either through direct contact or from eating food or water that's contaminated with feces that contain salmonella. So to help prevent salmonellosis in your backyard bird feeders, cleaning the feeders, taking them down, cleaning them with bleach and water. Wear gloves, because salmonella is something that people can get. So you want to wear gloves. You want to wash it in a bucket. Don't wash it in your kitchen sink. And then wash your hands really well afterwards. If there's an outbreak and you've got sick and dead birds at your bird feeder, then you'll want to take the feeder down, clean it, and leave it down for a couple of weeks so the birds aren't all congregated in that spot. You also want to store your store your bird seed in rodent-proof containers, because mice and rats and animals like that can carry salmonella bacteria and shed it in their droppings. And so you want to minimize that happening. And the third disease that I want to talk about a little bit was sylvatic plague. So the animal on the left is a black-footed ferret. This on the right is a prairie dog. And prairie dogs are hugely important for grassland ecosystems in the West. A lot of animals depend on prairie dogs or on their burrows, particularly black-footed ferrets. That's the main thing that they eat are prairie dogs, and prairie dogs are very susceptible to plague. And so there have been plague outbreaks in the West that, again, have been very devastating. We can lose up to 90% of the prairie dogs in an area. And so when there's a crash in the prairie dog population, then it's very bad for black-footed ferrets and the other animals that depend on them. And so what happened was one of our folks at the center, Dr. Toni Roque, and the folks in her lab and a whole plethora of partners, state, federal, tribal, and non-government organization partners, worked together to create a vaccine. And they put this vaccine in baits, and they had to figure out what flavor prairie dogs would like to eat. And it turns out they like peanut butter. And so these are peanut butter flavored baits. And this picture here shows a prairie dog holding and eating a bait. And it's a way to get the vaccine into the prairie dog. It's safe for them. It's safe for other animals. They broadcast these baits. They use it in multiple states. And it was a way to, again, try and work on disease control, disease management at a population level, because trying to keep the prairie dogs healthy helps keep the whole ecosystem in that area healthy. And that really leads us to the next point, the importance of having healthy wildlife. So why care? Why spend all of this? Why spend federal tax dollars? Why spend all this time and effort? Why should we care about wildlife health? Well, one thing is just the sheer beauty of natural areas and wild places. These are public resources, they're in the public trust. And so we have a collective responsibility then to manage these resources for future generations. In addition, being in nature is very healthy. Research studies have shown that there's a very good benefit to your mental health from spending time in nature. And some doctors, like the Missouri Department of Natural Resources, has been working with physicians in the state, and they are looking at prescribing time in nature to their patients. So there's a human health benefit there. And there's a lot of activities, that happen on public lands, camping, fishing, bird watching, hiking, and all of those things contribute not only enjoyment, but contribute to the economy as well. So the National Park Service periodically takes a look at that. And in 2021, so during the pandemic, they still had 297 million park visitors. And collectively, when they looked at the impact of those visitors to the surrounding areas and all of the different activities that they're doing, it added $42.5 billion to the economy. And that's just for the national parks. That doesn't include national wildlife refuges or state parks, county parks, or all the other ways that people get out and enjoy state lands. So healthy people, healthy environment, and healthy animals, they all go together. They all are interdependent on each other. So we should care about healthy wildlife because that helps us to be healthy, and healthy wildlife helps the environment to be healthy. And in turn, we help each other to be healthy. So there's a few resources in case you want to know even more about wildlife health. I wanted to share some resources with you. So if you're concerned about wildlife disease exposure or illness, either in yourself or your pets or your livestock, you should talk to your doctor or your veterinarian. If you're seeing sick or dead wildlife, or you have wildlife questions or issues, you should talk to your state natural resource folks. They often have wildlife health sections with wildlife health veterinarians, and they can help you. They have the legislative authority, and they have all the permits that are needed because you have to have special permission and permits to handle wildlife, and they already have them because they manage all of that. So you can find a list at fishwildlife.org, and you can find your state agency there. If you want to know more about the National Wildlife Health Center, you can visit our website. And if you want access to over 40 years of wildlife mortality event data, then check out whispers.usgs.gov. And there's a plethora of information about white nose syndrome in bats on whitenosesyndrome.org. Including the map that I showed you of all the different locations. That's actually a time-lapse map. And so you can see how it swept across the country from east to west over the years. And as I said, millions and millions of bats have been affected. Oh, and one other thing I wanted to point out when I talked about the economic value of the activities of people, I also meant to point out the economic benefit of wildlife, using bats as an example, because bats are pollinators for one. In fact, they're important there's a plant that's involved in the making of tequila, and bats are an important pollinator for that plant. But they also eat a tremendous amount of insects, and they estimated that each year, all of the insects that bats eat save farmers $3.7 billion. So there is an economic benefit to having healthy wildlife as well. So if all of this has you interested in being involved in wildlife conservation on a large scale, then you should check out usajobs.gov. And it lists Department of Interior jobs there and for the different agencies. And it's not just, scientists, researchers jobs. There's all kinds of jobs there from all kinds of professions. So you should definitely check that out. Well, I hope that next time you see a headline about a wildlife disease die-off, that you think about all of the things that are happening behind that headline, all of the processes that are going through, the communications and the collaborations that are going on between the state and federal partners, the tribal and non-government organizations. Check and see if they mention the USGS National Wildlife Health Center. We often get interviewed about different wildlife diseases. I hope you have a better understanding of the diagnostic process that we use to determine the cause of death in wildlife, and some of the things that we can do for disease response to see if there are ways to manage or control or prevent disease outbreaks. And lastly, I hope that I've helped you to understand why wildlife health is important and why we should all care about wildlife health. So thank you very much for this opportunity to talk about the center. I could talk about it all night, but I will stop here and see if there are any questions.
Thank you, Kim, so much. I could listen to you talk all night. I'm sure others out there could too, but that was great. And, you know, getting that behind the scenes look at how you and your colleagues handle these disease investigations from the beginning stages through the end was just fascinating to listen to, and all your photos. And I have to say, I can't help but comment on one of the first slides. There was a photo of you holding a really large sloth that I'm incredibly jealous. Oh, well, anyhow, so I've been monitoring all the questions that have come through. We've got a lot of great ones.
Okay.
And just a reminder, if anybody has one to ask Kim, go ahead and click on the Q&A panel. It's the question mark icon in the upper right-hand corner of your screen. So let's get through some of these. Okay, so the first one here, Kim. Anonymous, they say, great talk. How many birds or animals might be in your research facility at any given time?
Well, that's a good question. So everything that comes into the center to the diagnostic lab is already dead. When we request people ship carcasses to us, we are generally requesting anywhere from like 2 to 5 be sent in. So maybe we may have, I'm just guessing, in a week, we may be getting in, say, 30 different specimens over time, just roughly. It waxes and wanes. So there are times like we're doing a lot of work with high path avian influenza, and so we may get an influx of specimens in. So it can vary a lot. Now, I should say that the smallest things that come into the center are tadpoles, and the largest things are probably red wolves, gray wolves, and sea otters. Anything bigger than that doesn't really fit in a cooler very well. And so sometimes when we were doing, we had a scientist that was doing a lot of work with frogs and tadpoles. And so he may get in like 50 in a shipment. But generally we're requesting people send, 2 to 5 specimens to us. And we work through, like I said, the state and federal biologists. We don't have things coming into us from the public. We have things, they're routing them through the state and federal biologists. But we've received, last year, we received over 116 different species of birds over the course of the year.
Wow. Yeah, no, that definitely illustrates the amount of work you guys do there. Okay, another question from Anonymous. Do you ever use volunteers to help with your studies? How would someone go about finding these opportunities?
So the volunteers that we have, we do occasionally have volunteers at the center. We sometimes have vet externs that come to the center as well. The volunteers they're not necessarily involved with studies. They may be doing other things. We had a volunteer that worked in our library for a while there, I think, helping the librarian out, for example. But if you're looking for ways to be involved with wildlife, again, I would say check with your state natural resource agency, and because they are going to know who is doing, say, restoration work on the state park. So there may be things locally that you could be involved in, either habitat restoration or things like that, because there definitely are opportunities there.
Okay, great. And I did earlier when you mentioned USA Jobs, I did put that link in the Q&A
Okay.
for folks. And I just put a link on your National Wildlife Health Center website. There's also some more information on job opportunities. So that is in the chat Q&A. Okay, so another one here. I think you covered this, but they say, I've been noticing more dead birds in my area recently. Would I report that to your group or someone local?
Someone local. So if you go to the link that we shared, fishwildlife.org, and you can look up your state, and it'll have the link there for your state natural resource management agency. And I say that you want to contact your state because they are right there. They are going to be more familiar with what's going on in your area, and they have all the permits and everything that would be needed if they need to collect carcasses and send it into a lab. I also meant to say we're not the only lab around that does diagnostic work, but we really are the only lab at the federal level that is doing the type of work that we do. And one of the benefits is that we can see things then across all 50 states.
Here's a question from Larry. He says, I heard that the latest avian flu, the one wiping out our egg supply, might be crossing over to mammals. Do you study disease crossover from animals to humans?
So we do look, diseases that can infect animals, diseases of animals that people can get are called zoonotic diseases. And we do work with some zoonotic diseases. We tend to do that in partnership with say the public health folks. So we're looking at maybe the disease and in animals, and then the public health folks would be looking at the human side of that.
Okay, got it. I'm just scrolling down here. Here's another one from Anonymous. Did the USGS do any studies or consulting relating to coronavirus?
Yes, we did, actually. So we were involved in looking at the SARS-CoV-2 in wildlife. There was a concern as to, like, is it something that could get into bats or not? Doesn't seem to be. But yes, there are folks at the center, our virologist that has been involved. He has been involved with looking at SARS-CoV-2.
Okay. Another one from Anonymous. What is the current range of white nose syndrome? Have any affected bats been found in California?
Actually, if I can, let me see if I can get back to that slide for you. So hang on. I'm gonna have to, click back through. So the whitenosesyndrome.org has that, there it is, hang on, there. So yes, there's some, so the red circle is showing that there's, it looks like it's presumed, like they have maybe, it looks like they may have detected it. It is not confirmed as positive according to this, and this is going through 2022. So I don't know if there's something in these last few months. So I don't think they have confirmed bats with the fungus, but they may have detected. It looks like here they PD presumed. So there's some evidence there, but it's not confirmed yet in bats, unless, as I said, unless there's something in these past few months of 2023 that hasn't been updated on the map. Yeah. Okay,
great. And we did give a link to that map as well in the chat if they want to go back and look. Another one here. Are prairie dogs attracted by the smell of the peanut butter or the color of the bait?
Oh, that's a good question. That's a very good question, and I don't know the answer to that. I will have to ask Dr. Roki. It could be a combination of both because it is so brightly colored. Yeah, that's That's a very good question.
Let's see. Another one here. Are there any mitigation measures for the bat disease?
Well, there's a lot of things that are being studied right now. So the vaccine was one, trying to understand if it's possible to make a vaccine that would be efficacious and you could get it to enough bats and things like that. They're also looking at whether or not there are things, because it's a fungus that lives very well in the environment within the caves. And so they're looking at, are there things that we can do to alter the environment? Because to interrupt a disease cycle, you have to break the link somewhere in there. You either have to break the link between the host and the environment, or the agent in the environment, or the host and the agent. If you can interrupt that cycle of those three things, then you'll stop the disease process. It's just very challenging for all of the reasons that I said. You are working with populations, they're tiny, they are tucked back into areas. In fact, Aeolus Cave in Vermont, for the longest time, they thought it was not a very big cave. And some experienced cavers then found that there was a crevice between two rocks that they could kind of wiggle in, and they found that there was a longer passage that then got them to a big chamber, and that there were thousands of bats there. So that's how they ended up understanding that this was the largest bat colony in the Northeast, And so some of the places that you would have to get to the bats to try and help to treat them are quite challenging to get to. Or the mine that we were in with the really steep, I mean, that was very big and steep and difficult to maneuver in. So sometimes there's the ability to maybe do something hypothetically, but practically it can be quite challenging. So, but we don't want to keep losing millions of bats. So there is a lot of work by a number of different partners looking at what is feasible to do. Like, are there things that are reasonable to do? And can we be effective enough to make an impact on this disease, minimizing this disease?
Gotcha. And while you're on that slide, this next one, I think they might be testing if I can pronounce this word I'm about to say, so bear with me. Anonymous says, thanks for that white nose syndrome map. What does it mean in the areas where pseudogymnascus? Thank you.
It's really hard. Pseudogymnascus destructans. So what does it mean? So there's
where it's presumed, the person says, yeah.
Okay, so I think that they are perhaps detecting the fungus, because I know that there are some states where they have detected the fungus, but they haven't necessarily detected bat morbidity, sick or dead bats. And so I believe that is the case there, that they may have detected the fungus, but not the disease. And that's getting back to what I was talking about pathogen detection versus disease detection. So I know that there are states where they have found the fungus, but not found the disease yet.
Okay, great. And thank you for helping me with that.
Yeah, it's a tricky one. I was hoping I would not have to say it.
You're using me. It looks like we've got maybe 3 more here. This person's asking, how did the National Wildlife Health Center end up under the U.S. Geological Survey?
Yes. So we started out with Fish and Wildlife Service. And then in the early 90s, there was Department of Interior decided to pull the research from their different agencies, that Fish and Wildlife Service, National Park Service, and put the research into its own group. And the reason they wanted to do that is because they wanted to separate out the science part from the enforcement part. So in USGS, we don't make the rules, we do not enforce the rules, we do the science. And so we can't tell people what to do. We can tell you, based on the best that we know from the science, that XYZ thing may happen. Like if you do this, then this is likely to happen. But we can't make anyone do that. So that was really kind of the intent of trying to consolidate the research. And also they wanted the research to be more collaborative as well. And so they put it into its own group. And then there was some politics that happened. And eventually we were moved over into USGS, which I think is really a good fit because, as I said, USGS is the science arm, and we do the science. And so that's how we ended up in USGS after starting in Fish and Wildlife Service.
Nice. And I do think a lot of folks just kind of think of USGS sometimes as earthquakes and volcanoes, but yeah, that's why we love to do talks like this, because there's just so much more to it. So okay, two more here. This one's from Art. They say there's been a problem of canine distemper reported with gray foxes in the Palo Alto Baylands. Have you investigated this?
I don't think we've had specimens come into the center, but I'm not entirely sure about that. But I imagine it's probably being handled at the state level through the state diagnostic lab there. I don't remember any recent submissions from that area for that disease.
Okay. Last one, aside from all the wonderful talk comments and all those, I won't go through all those, but last one, let's see, few sentences. They say, thanks for a wonderful talk. I am curious about the idea that pathogens are also natural components of animal populations and ecosystems. Clearly, they can get out of balance, often due to human activities. So how do you determine the natural balance of pathogens and diseases that should occur and whether or not they need to be addressed?
Oh, that's a good question. Okay, so there are a number of different things that are in the environment. So for example, if a lab person were to come in and take a swab of the back of my hand, they could grow bacteria, for example. But finding that bacteria on my skin doesn't mean I have that disease. It just means that they found that organism. So there are a number of organisms all over our environment that do not cause a problem for us or for people. Now, sometimes what happens, if I had a cut on my hand, let's say I'm gardening, I'm playing in the dirt, there's no problem with that. If I had a cut on my hand and I'm gardening and playing in the dirt, and those organisms in the dirt can get down, past my skin, then that's when you can start having a problem because it's gone past that barrier and it's causing an infection. So if it's not interfering or interrupting normal body function, then oftentimes it's not a problem. It's just something that we have detected. So when we do cultures, say on animals, they detect all kinds of bacteria. For example, you can culture the intestines and come up with all kinds of bacteria, but some of those, they're supposed to be there. They're healthy. They're part of the ecosystem of that animal, and they're not causing a problem. So the focus really is on those organisms that are interrupting and interfering with normal body function and are causing some sort of problem. So those are the ones that we're focusing on.
Yep, makes sense. And two more did squeak through. One's
Sure.
From Virginia. Your presentation was most interesting, great pictures and information. And this will be the last question. Anonymous says, thanks for an incredible talk, Dr. Miller. What does the next 5 years look like in this pathogen landscape?
Oh, these are great questions. Well, so the diseases that we've been seeing, these new and emerging diseases, a lot of them have a wildlife component. And I suspect we will continue seeing new and emerging diseases on the landscape. And part of that is there's a number of reasons, I think. Certainly, as populations expand, then you get more of an interaction between people and wildlife, for one. So habitats tend to get more fragmented and broken up, which puts stresses on wildlife, stresses can be additive. So when you look at weather stress, when you look at habitat stress, if you look at food resources decreasing, those are all cumulative stresses for animals and can make them more susceptible to disease problems. So I think that we will continue seeing new and emerging diseases, or seeing common diseases kind of flare up. So there are some diseases like avian botulism that we see every year, every summer. So I anticipate that we will see, more and perhaps larger, longer outbreaks just because of the way that disease works. And when you have areas that are experiencing drought, for example, and the water levels are decreasing, it can create the conditions that are conducive to botulism, because botulism is a spore that can be dormant for a long time, and then under the right conditions, it can become active and produce the toxin. And some of the things it needs are an anaerobic or no oxygen environment, a protein source, and warm temperatures. And so, for example, if you have increasing summertime temperatures, you have decreasing water levels, you're kind of creating the conditions for avian botulism outbreaks to occur, for example. So I think we will continue to see wildlife diseases, some new and emerging. So stay tuned, and you can visit our website. If you look at our website, you will see, especially, you can get to over 40 years of wildlife mortality information. There's a map there that shows you recent reports that partners have shared with us about sick and dead wildlife. So, and you don't have to sign in or anything. You can just visit the whispers.gov site and see the map of recent reports.
Well, great. I'm glad that last question squeaked through. It was kind of the perfect way to round this out. So we really can't thank you enough for your talk tonight. It was fascinating. We got great questions, and we also want to thank everyone out there for joining us for the lecture. And in case you want to watch Dr. Miller's talk again or share it with others, her lecture will be available in about a week for on-demand viewing on our website, which is www.usgs.gov/PLS. On that website, you can find videos from our past public lectures spanning the past 20 years, and our lecture schedule for the rest of 2023. And we've got a lot more great talks lined up, including talks on our water alert system, landslides, snowpack dynamics, how to create on-demand topo maps, and a lot more. And also, if you want to be part of our mailing list, so you'll get an email twice a month letting you know what the lecture of the month is, you can go ahead and send us an email to sign up at wmcesic@usgs.gov, and we can add you to our list. And again, we hope you'll join us next month on March 30th at 6 p.m. Pacific time for Ilse Kuffner's talk on Coral Reefs in Crisis, Science to Guide Reef Restoration for Ecosystem Recovery. And until then, thank you again to Dr. Miller. Thank you to all of you out there, and we hope you have a good night. Goodbye.
Thank you.